Hello everyone and welcome to the new session of the lecture series, and today's topic is respiration in plants, and today we'll be discussing and solving all the NEET previous year questions. Okay, so let's start with the first question. So here they are asking how many times decarboxylation. Okay, decarboxylation means what happens during decarboxylation? There is a release of carbon dioxide. Okay, there is a loss of carbon dioxide. So they are asking how many times decarboxylation occurs during each TCA cycle. Okay, each means one TCA cycle. Okay, because TCA cycle, also known as the Krebs cycle, takes place twice because two molecules of pyruvate are formed. Okay, and correspondingly two molecules of acetyl coenzyme are formed. So actually Krebs cycle or TCA cycle take two times, but they are asking about each TCA cycle. So what happens when isocitrate, okay, is converted into alpha ketoglutarate? Okay, then what is happening here? There is a loss of carbon dioxide. Okay, so this is the first decarboxylation. Okay, this reaction is catalyzed by the enzyme isocitrate dehydrogenase. Alright, isocitrate dehydrogenase. And what else is happening here? There is conversion of NAD into NADH. Okay, now what happens in the next step? So isocitrate is a 6-carbon compound and alpha ketoglutarate becomes 5-carbon compound. Now second decarboxylation takes place when alpha ketoglutarate, okay, is converted into succinyl coenzyme A. Okay, here also there is a release of carbon dioxide. Okay, and again what is happening? NAD is being converted into NADH. Alright, this reaction is catalyzed by the enzyme alpha ketoglutarate dehydrogenase. Okay, alpha ketoglutarate dehydrogenase. Again, succinyl coenzyme A is a 4-carbon compound. Okay, and you can say coenzyme A is also getting added here. So what should be the answer? Answer should be twice. Okay, 2 times. Now coming to the next question, fatty acids are connected with the respiratory pathway through, okay, these are the options: acetyl coenzyme A, alpha keto, glutaric acid, dihydroxyacetone phosphate, and pyruvic acid. Alright, so fatty acid they are asking. So fatty acids, okay, they form acetyl coenzyme A. Okay, now this acetyl coenzyme A can enter the Krebs cycle. Okay, acetyl coenzyme is also formed from pyruvate. From where this pyruvate comes? This comes from glucose. Okay, glucose during glycolysis, okay, is converted into pyruvate. Pyruvate, oxidative decarboxylation takes place, converted into acetyl coenzyme A. So here they are asking fatty acids are connected to the respiratory pathway. This is the respiratory pathway here. Okay, so what should be the answer? Answer should be acetyl coenzyme A. Okay, option A. The next question is, given below are the two statements. Okay, one is labeled as assertion A and the other is labeled as reason R. Okay, so let's start with the assertion. So here they are saying ATP is used at 2-step in glycolysis. Okay, so this is true. Yes, this is true. Alright, and second they are saying first ATP is used in converting glucose into glucose 6-phosphate. Yes, and second ATP is used in the conversion of fructose 6-phosphate to glucose 1,6-diphosphate. Alright, so these are the options. You have to choose the correct answer from the options given below. Now we know that statement okay, assertion and reason both are true and reason is the correct explanation of the assertion. Okay, let's talk about glycolysis where ATP is used. Okay, first they are saying when glucose is converted into glucose 6-phosphate. Okay, here one ATP is utilized. Okay, and it is converted into ADP. Okay, this reaction is catalyzed by the enzyme hexokinase. Second ATP consumption is when fructose 6-phosphate, okay, is converted into fructose 1,6-diphosphate. Okay, this is also a phosphorylation step. Okay, and what is happening here? ATP is being used and getting converted into ADP. Alright, so these were the statements. Okay, so correct answer is option D. Now coming to the next question, which of the following combination is required for chemiosmosis? Okay, what was chemiosmosis? Formation of ATP, okay, by the breakdown of the proton gradient. Alright, so these are the options: membrane, proton pump, proton gradient, NAD. Okay, so this is not true because NAD synthase is not involved. Alright, in chemiosmosis. Second option is proton pump, electron gradient and ATP synthase. Again, it is missing the membrane because membrane is also required. Option C is proton pump, electron gradient and NADP synthase. This is also not true. NADP synthase is not involved. Now, fourth option is membrane, proton pump, proton gradient and ATP synthase. So this option is correct. Alright, so for chemiosmosis, membrane is required. So the electron transport chain, okay, the complexes of electron transport chain are present in the inner mitochondrial membrane. Okay, they are present in the inner mitochondrial membrane. And what happens? And as the electrons flow downhill, okay, from one complex to another, okay, so what happens? A proton gradient is created with the help of proton pump. Okay, so proton pump, what does it do? It helps in the build-up of the proton gradient. Help in the build-up of proton gradient. Alright. So here complexes are present: complex 1, complex 3, complex 2, then we have complex 4, okay, and then we have the ATP synthase, which is known as complex 5. Okay, as the electrons flow here downhill, okay, protons from the matrix, okay, they are transported to intermembrane space. Okay, and what happens? These protons get initially accumulated in the intermembrane space, and later on, okay, they flow back to the matrix. Okay, and what happens here? ATP is generated, okay, from ADP and inorganic phosphate. And what is, okay, how ATP is generated? With the help of ATP synthase, which is known as the complex 5. Okay, so correct option is option D. Membrane is also required because proton gradient forms across the membrane. Alright. Now, here we have to match the list 1 with the list 2. Okay, so this is list 1. Oxidative decarboxylation. Okay, what happens? Oxidative decarboxylation, glucose is converted, pyruvate is converted into acetyl coenzyme A, and the enzyme involved here is pyruvate dehydrogenase. So A should be second. Okay, then we have glycolysis, is also known as EMP pathway, Emden-Meyerhoff-Parnas pathway. Okay, EMP pathway. So option B should be fourth. Now see, oxidative phosphorylation. Okay, electron transport system. Okay, it is related to electron transport system. Then tricarboxylic acid, also known as Krebs cycle. Okay, it is related to the citrate synthase, which is involved in the formation of citrate. So option D should be 1. Okay, so let's look at the options. These are the options. Okay, then what should be the correct answer? Correct option is C. Alright. Now, next question is, malonate inhibits the growth of pathogenic bacteria by inhibiting the activity of. Okay, so let's talk about malonate first. What is malonate? Malonate has a structure similar to succinate. Okay, malonate has a structure that is similar to succinate. Okay, so that's why it acts as a competitive inhibitor. Okay, it acts as the competitive inhibitor of which enzyme? Succinic dehydrogenase. Okay, or succinate dehydrogenase. Okay, and it's compete with the succinate. Okay, it's compete with succinate to bind to the active site of succinic dehydrogenase. Alright, succinate dehydrogenase. What is the role of succinate dehydrogenase? In Krebs cycle, it converts succinate into fumarate. Okay, and what is happening? FADH2 is produced from FAD. Okay, so what should be the answer? Answer is option D. Malonate inhibit the growth of pathogenic bacteria by inhibiting the activity of succinic dehydrogenase. Now next question is, number of time decarboxylation of isocitrate occurs during, again, single, they are asking, single TCA cycle. Okay, then what should be the answer? Again, this is straightforward question. Answer is option C, 2. Why? Because let's look at the TCA cycle. What happens? Initially we have citrate gets converted into isocitrate. Okay, citrate is a 6 carbon compound. It isomerizes into isocitrate. Isocitrate is converted into alpha ketoglutarate. okay, which is a 5 carbon compound. Here carbon dioxide is released. Okay, this is decarboxylation step. And what happens? This alpha ketoglutarate is further converted into succinyl coenzyme A. Okay, this is second decarboxylation. Okay, so there are two decarboxylation. This is first decarboxylation and this one is second decarboxylation. Okay, so number of time decarboxylation of isocitrate occurs in single TC cycle is 2. Okay. Now, what is the net gain of ATP when each molecule of glucose is converted into 2 molecules of pyruvic acid? So here they are talking about the glycolysis. Okay, net gain also they are asking about net gain. Okay, so what happens during the preparatory phase? During the preparatory phase, what happens? 2 ATP are used, okay, are consumed. Okay, then later what happens? 4 ATP are produced. So net gain is 2 ATP. Alright. So what happens when glucose, okay, is converted into glucose 6-phosphate? 1 ATP is used here. Okay. Now when later what happens when fructose 6-phosphate is converted into fructose 1,6-bisphosphate or diphosphate, again 1 ATP is used. Okay, you can write cross means it is being used. Then later what happens? ATP is produced later. In which stages? When 1,3-diphosphoglycerate is converted into 3-phosphoglycerate. During this what happens? ATP is produced. Okay, now we have 2 molecules of diphosphoglycerate means 2 ATP are produced. Okay, and later what happens? When phosphoenol pyruvate is converted into pyruvate. Okay, then also 2 ATP are produced. Okay, why? Because there are 2 molecules of phosphoenol pyruvate. So in total 4 ATP are produced, 2 ATP were consumed. So net gain is 2 ATP. So what should be the answer? 2 ATP. The next question is, what amount of energy is released from glucose during the lactic acid fermentation? So lactic acid fermentation takes place without oxygen. Okay, there is no use of oxygen. And what happens? Pyruvate, which is produced at the end of glycolysis, is converted into lactic acid. Okay, so the amount of released energy is very less. Okay, so correct option is option D. Okay, less than 7%. Okay, so during anaerobic respiration such as lactic acid fermentation, okay, the amount of energy in the glucose molecule, okay, not all of them are converted into ATP, but only 7% of the energy is used, okay, for the formation of ATP, okay, and not all. Now, which of the following statements is incorrect? Okay, so these are the statements. Okay, first one is oxidation reduction reactions produce proton gradient in respiration. Okay, so this is true, but we have to select the incorrect statement. Okay, this is true. Option B is during aerobic respiration, role of oxygen is limited to the terminal stage. Yes, so they are talking about the terminal. Okay, oxidation here. Okay, oxygen accepts electrons and protons and gets converted into water. Option C is in electron transport chain, one molecule of NADH give rise to 2 ATP, whereas FADH2 give rise to 3 ATP. So this is not correct, because we know that NADH, one NADH corresponds to 3 ATP. Okay, and one FADH2, okay, gives us 2 ATP. Okay, this is incorrect. This is also correct. ATP synthesized through complex 5, which is known as ATP synthase. So correct option is option C. Okay, so answer is option C. The next question is number of substrate level phosphorylation in 1 turn of citric acid cycle. Okay, so substrate level phosphorylation, what is? Okay, leads to the formation of ATP or GTP. Okay, so where does this substrate level phosphorylation take place? When succinyl coenzyme A is converted into succinate. Okay, here what happens? GTP is formed from GDP. Okay, so this is the substrate level phosphorylation taking place at this step. Substrate level phosphorylation is taking place here. Okay, and what is substrate level phosphorylation? Formation of ATP or GTP by the transfer of the phosphate group. Transfer of the phosphate group from a high energy compound okay, to ADP or GDP. Okay, this is GDP. So what should be the answer? Answer is 1. The next question is, what is the role of NAD in cellular respiration? Okay, so options are, it is the final electron acceptor in the anaerobic respiration. Okay, but what happens during anaerobic respiration? NAD is produced. Okay, NAD is regenerated. This is regenerated. Okay, option B is it functions as an enzyme. Okay, which is wrong because it is a coenzyme. It functions as an electron carrier. Okay, so this is true. It acts as an electron carrier. Okay. Now, option 4th is it is a nucleotide source for ATP synthesis, which is again not correct. So, what is the role of NAD? It functions as an electron carrier. Okay. If we talk about the option A, why it is not correct? Because the electron acceptor in anaerobic respiration, like lactic acid fermentation, is okay, acetaldehyde. Okay, we know that in lactic acid fermentation, what happens? Oh, sorry, in alcoholic fermentation, not lactic acid fermentation. During alcoholic fermentation, pyruvate, it's converted into acetaldehyde first. Okay, this acetaldehyde, this is a electron acceptor. Okay, and not NAD. And acetaldehyde finally converted into ethanol. Okay, and carbon dioxide is released as a byproduct. So correct option is option C. The next question is, which statement is wrong for the Krebs cycle? Options are, there is one point in the cycle where FAD is reduced to FADH2. This is true. Okay. Option B, during conversion of succinyl coenzyme A to succinic acid, a molecule of GTP synthesized. This is also true. A cycle starts with the condensation of acetyl group, okay, with the pyruvic acid. So this is wrong because there is condensation of acetyl coenzyme with oxaloacetic acid, okay, which leads to the formation of citrate. So this option is correct. Now next option is there are 3 points in the cycle where NAD is reduced to NADH. Okay. So what is the correct answer? Answer is option C. The cycle starts with okay. So this is correct answer is option C. Okay, because here oxaloacetic acid is involved. All the other options are correct. And what we have to mention? We have to mention the wrong statement. So next question is, all enzymes of the TCA cycle are located in the mitochondrial matrix except one which is located in the inner mitochondrial membrane in eukaryotes and in cytosol in prokaryotes. So we have to name this enzyme. So these are the options. Okay. So they are asking about the enzyme present in the inner mitochondrial membrane. So now we know that in the inner mitochondrial membrane, we know that the complexes of the electron transport system are present. Complex 1, which is known as NADH dehydrogenase. Then we have complex 3, which is cytochrome BC1. Complex 2, okay, which is known as succinate dehydrogenase. Okay. And then we have the complex 4, which is known as the cytochrome C oxidase. So, succinate dehydrogenase, okay, which is complex 2. Succinate dehydrogenase is present in the inner mitochondrial membrane. Okay. So, it connects the Krebs cycle with the electron transport system directly. So, correct answer is succinate dehydrogenase. And it is also present in the cytosol. Now, next question is, the overall goal of glycolysis, Krebs cycle and the electron transport system is the formation of, this is straightforward answer. Answer should be ATP in small stepwise units. Why? Because during glycolysis, what happens? ATP is produced. Okay, net gain is 2 ATP. Okay, then during Krebs cycle, what happens? GTP is formed, which is equivalent to 1 ATP. Okay, 1 GTP is equivalent to 1 ATP. Okay, Krebs cycle occurs 2 times, so 2 ATP are formed. Okay, again during electron transport, NADH, okay, NADH is oxidized and ATP is produced. Okay, during electron transport system. Okay, so ATP is synthesized in small stepwise units, okay, and not in one large oxidation reaction. Okay, and nucleic acid is also not true. Sugars are also not true. So what should be the correct answer? Option D. The next question is, during which stage in the complete oxidation of glucose are the greatest number of ATP molecules formed during, okay, from ADP? Okay, so what happens during glycolysis? We know, so they are talking about the complete oxidation, means they are talking about the aerobic respiration. Okay, and what are the steps involved in during the complete oxidation? First is glycolysis. Okay, what happens during glycolysis? ATP is produced, 2 ATP are produced, plus 2 NADH is produced. This NADH can go to electron transport chain, okay, and lead to the generation of ATP later, okay, because this is the source of electron, okay. This is oxidized, okay, electron move from one complex to another downhill and proton gradient is generated, and finally breakdown of proton gradient leads to the formation of ATP. And then again during Krebs cycle, what happened? 6 NADH are produced, okay, as well as 2 FADH2 are produced, okay. These can also go to the, okay, will go to the electron transport system, Again, before Krebs cycle, we have oxidative decarboxylation. And what happens during oxidative? Again, 2 NADH are produced. Okay, so this can also go to electron transport chain. So what should be the answer? Greatest number of ATP produced will be in the electron transport chain. Okay, because all the NADH and FADH2 will be going to the in the electron transport system and generate ATP. Now conversion of glucose to glucose 6-phosphate. The first irreversible reaction of glycolysis is catalyzed by, this is straightforward answer, hexokinase. Okay, hexokinase involved in the conversion of glucose to glucose 6-phosphate. Okay, and what does it do? It do phosphorylation. Okay, this is the first step of glycolysis known as phosphorylation. And ATP here is converted into ADP. So option D is the correct answer. Now, next question is, which of these statement is incorrect? We have to select. Okay, so options are, glycolysis operate as long as it is supplied with NAD plus that can pick up the hydrogen atom. Okay, so this is true. Now option B is, enzyme of TCA cycle are present in the mitochondrial matrix. This is also true because TCA cycle takes place in matrix. Now option C is, oxidative phosphorylation takes place in outer mitochondrial membrane. Okay, so this is not true because it takes place in the inner mitochondrial membrane. Okay, because electron transport system, the complexes of electron transport system are present in the inner mitochondrial membrane. Okay, now option D is, glycolysis occurs in cytosol. This is also true. So this option is incorrect. So answer should be, okay, oxidative phosphorylation takes place in the outer mitochondrial membrane. The next question is, oxidative phosphorylation is, they are asking. Okay, so option first is, formation of ATP by transfer of phosphate group from a substrate to ADP. So this is not true. Now next option is, oxidation of phosphate group in ATP. Okay, this is also not true. Option C is, addition of phosphate group to ATP. Okay, this is also not true. This usually takes place in the substrate level phosphorylation. Okay. Now option D is, formation of ATP by energy released from electrons removed from during the substrate oxidation. So this is true. So what happens during oxidative phosphorylation? The energy, okay, released by the or during the oxidation reduction reaction. Okay, one molecule is oxidized, other molecule is reduced. Okay, which molecule is oxidized during oxidative phosphorylation? NADH, which is a reduced coenzyme, is oxidized to NAD. Okay, so energy released by the oxidation reduction, okay, oxidation reduction reactions is utilized for the formation of ATP. Okay, so option D is the correct answer. Now coming to the next question, which of the following biomolecules is common to respiration mediated breakdown of fat, carbohydrates and proteins? So we know that respiratory pathway is interconnected. Okay, and what happens? Suppose we have fats here, then we have carbohydrates, and then we have proteins. Okay, fats will break down and they will form acetyl coenzyme A. Carbohydrate will first produce glucose. okay, which will convert into pyruvate, and pyruvate with oxidative, okay, will undergo oxidative decarboxylation to form acetyl coenzyme A. Now again, proteins, okay, will break down into amino acids. Amino acid now can form pyruvate, okay, they will get converted into pyruvate, and pyruvate can then form acetyl coenzyme A. So acetyl coenzyme A, which is formed, can enter the TCA cycle now, okay, or the Krebs cycle. Alright, so which is the molecule which interconnect? Okay, answer should be option D, acetyl coenzyme A. So this was the end of this lecture. We'll continue with rest of the questions in the next lecture. Thank you for listening.